So today I'm going to be talking about learning biological networks, or more specifically, techniques for estimating high-dimensional graphical models. And this is something that actually is really relevant in a lot of areas of biology nowadays, as we are using assays that collect more and more measurements on a single tissue sample. And so I'm going to be talking about a few different projects. And so just briefly, this work is joined with a number of collaborators, some of whom are at Stanford, one of whom is at Stanford but is on his way to UW, and then most of the other people are at UW or at the Fred Hutchinson. So my talk has three parts. I'm going to start by an overview of what I mean by statistical network estimation. And then I'm going to talk about how we can actually use some of the statistical machine learning methods that I work on in order to try to understand biological networks, and in particular, recent work on how we can jointly estimate multiple networks in order to try to really understand the difference on a network level between, for instance, normal versus tumor tissue. So I'm going to begin with an overview of statistical network estimation. And when I'm using the word network, as we're going to see, I have a very precise statistical meaning in mind, and so I'm really going to spend a lot of this talk explaining what I mean when I'm talking about a network. So we, of course, know that genes, proteins, and other molecules that we might be interested in don't operate alone. And instead, they operate as part of complex pathways or networks. And so we know that if we do an assay, for instance, measuring 20,000 genes in a tissue sample, we're going to need to somehow borrow strength across those genes or somehow pool those genes together, or really think of those genes as interacting molecules rather than as individual features. So we want a network understanding of our data, hopefully in order to obtain a better understanding of both normal and disease processes. And so today I'm going to be talking about a statistical approach for estimating biological networks, and this is known as Gaussian graphical models, and I'm going to explain what that means. So this is an example of what I'm referring to when I talk about a gene network or a protein network. And we can see that there's a lot of molecules involved, and it's pretty complex, and there's a lot of arrows indicating a molecule that's upregulating or downregulating another. And this is something that we're really interested in because it allows us to understand how processes are happening biologically. And traditionally, of course, in order to be able to determine a network like this, it would require a lot of experiments. And so the question is there sort of a more efficient way that we can make use of high throughput data, for instance, gene expression data, in order to be able to infer networks like this one in an automated way? So in particular, can we estimate networks using observational gene expression data? So we can get a lot of gene expression data from microarrays or from sequencing. And for instance, let's say we have a data set with 200 patients with, let's say, lung cancer. And we have 20,000 gene expression measurements per patient. And the question is, can we estimate the gene expression network? Can we understand how these genes are interacting in these tissue samples? So really, our goal is to be able to infer a network like this one, not on the basis of really labor-intensive experiments. Well, I'm a statistician, so I never have to do labor-intensive experiments. But the goal would really be to be able to infer a network like this one on the basis of gene expression data or sequencing data that can be obtained in a more efficient way than, for instance, going into the lab and testing every single edge in this network in order to see whether a given pair of molecules interacts directly. So can we find sort of a high throughput statistical way to analyze this high throughput biological data? So in order for this question to make sense, we need to, first of all, be clear about what it is that we mean by a network. So I'm going to be showing pictures like this, and in this picture, each node represents a gene. So here I'm talking about genes, but these don't have to be genes. These could be any sort of biological molecules of interest. And the idea is that some of these nodes are connected by edges. And if a pair of nodes is connected by an edge, then that indicates a pair of genes that are conditionally dependent. And so what that means is that it is a pair of genes that has a direct relationship, not an indirect relationship due to other genes, but rather a direct relationship. For instance, one gene acts on another. And this notion of conditional dependence has a really precise statistical meaning. So just to be totally clear, It doesn't mean that these two genes are correlated with each other. It doesn't mean that if one of their expressions goes up, then the other expression goes up, or if one goes down, the other goes down. What it means is that they have a relationship that cannot be explained away by the other genes in the data. So there has to be evidence that they act directly rather than indirectly. Even after you condition on all of the other genes, they still have a relationship. So that's what I mean if I talk about an edge between a pair of genes. And in contrast, some genes aren't connected by an edge, and this corresponds to a pair of genes that's conditionally independent. So these two genes, they aren't connected by an edge, but they might have correlated expression values. And in fact, they probably do, because as we can see in this figure, there's a third gene that's connected to both of them. So if we just think about correlation or a notion like that, we would erroneously think that there's a relationship between these two genes here that we would erroneously think that there's a connection between these two genes, but in fact there's not. They're conditionally independent. And so this is what I'm going to be talking about today, and this is what I'll be referring to as a network. So this is actually a pretty simple concept. Let's say that instead of 20,000 genes, we have 3 friends. And Mike and Sally are friends. Sally and Joe are friends. And so this is the network of friendship between Sally, Mike, and Joe. But it just so happens that Mike and Joe often show up at the same parties. And it's not because they're friends. It's because they're both friends with Sally. And so there's this indirect relationship between Mike and Joe that is due to Sally. So if we just think about correlations, or how often Mike and Joe and Sally happen to be at the same party, we might erroneously draw an edge between Mike and Joe. But we shouldn't, because the true relationship is like this. The only relationship between Mike and Joe is due to the fact that they're both friends with Sally. And so in the context of biology, this would correspond to the idea that we have 2 genes, or rather 3 genes. 2 of them are directly related to a third, and those 2 are not directly related to each other, but they tend to have correlated, let's say, expression with each other due to the relationship with that third gene. So going back to this friendship example, what would we actually want to do in this case? Well, we could collect some data. We could go around to 200 different parties, and at each party we could record whether or not Sally, Mike, or Joe attend. And so then our data matrix would be a 200 by 3 data set with 3 columns, 200 rows, and each element is a 0 or a 1. And on the basis of this data, we can compute a correlation matrix between Sally, Mike, and Joe's party attendance. And the question is, can we use this to recover their relationships? So our data looks like this. We have 200 rows, each corresponding to a different party. 3 columns, 1 for each of these 3 people, and each element is a 0 or a 1. It's a 1 if the particular person attended a particular party. And we can see that if Sally goes to a party, Mike or Joe or both tend to attend. And if Sally doesn't go, then maybe Mike doesn't go. So on the basis of this data, we can calculate correlations. And so this is a correlation matrix of party attendance. So it's a 3 by 3 matrix. And so we see that on the diagonal, the values are 1, because Sally's party attendance is perfectly correlated with Sally's party attendance, and so on. Mike and Sally have a correlation of 0.58. Joe and Sally have a correlation of 0.58, and Mike and Joe have a correlation of 0.33. So if we were to try to infer the underlying friendship network of Mike, Sally, and Joe by looking at this correlation matrix, we would infer that Mike, Joe, and Sally are all friends with each other. But they're not, and the reason is because we shouldn't be looking at correlations. This correlation between Joe and Mike of 0.33 is not due to a direct relationship between them. It's due to an indirect relationship due to shared friendship with Sally. So it turns out that the problem is that we're looking at the wrong thing. We're not actually interested in correlations. We're interested in inverse correlations. And so if you remember back to maybe a linear algebra class that you took a very long time ago, or that you did not take, if you have a matrix, most matrices, and in particular correlation matrices, are invertible. So corresponding to a matrix, there's another matrix called the inverse matrix. Not all matrices have inverses, but it doesn't matter. Correlation matrices do. So there's an inverse matrix, and you can just think of this as a mathematical construct. But it is a very useful mathematical construct because it has a really useful property, which is that if a particular element in the inverse correlation matrix is 0, then that corresponds exactly to Mike and Joe being conditionally independent. So again, in this example, Mike and Joe not being friends. So it turns out that if we're thinking about developing these networks, and again, whenever I say network in this talk, I'm talking about a particular type of network, which statisticians call a Gaussian graphical model. But under this type of model, if I want to know if there's an interaction between Mike and Joe or between gene A or gene B, I should not look at the correlations. I should look at the inverse correlations. And if I see a 0, then that tells me that they're conditionally independent. So it's actually, if you can, again, either reach back to your linear algebra or just believe me, it's actually pretty simple. All we care about are zeros and sigma inverse. So just to clarify this point, if you have a correlation matrix sigma, there's an associated inverse correlation matrix sigma inverse. If you hand me a sigma, I can't do it by hand, I'm sorry, but I can on a computer calculate the inverse correlation matrix sigma inverse. And so we might be tempted to look at correlations in order to learn the network structure, but it's going to be wrong, and we're not going to get the results that we want if what we really mean by an edge in the network is this notion of conditional dependence, which is to say, again, a relationship even after you take out the effect of all of the other variables that you've measured. So what we need to do is estimate sigma inverse, and in particular, we need a good way to estimate sigma inverse. And as we're going to see, estimating sigma inverse is actually pretty challenging, and that's the focus of a lot of my research. So that's kind of an overview of what I mean by statistical network estimation. And now I'm going to talk about my work on fast estimation of biological networks and how we can sort of translate these statistical concepts to actually try to solve problems that are of interest biologically. So now we're done with Mike, Sally, and Joe. I think. We might see them again on another slide. I guess we'll find out. We're almost done with Mike, Sally, and Joe, and we're moving on to, let's say, gene expression data, where we have a data matrix with 200 rows, each of which corresponds to a tissue sample, and let's say 20,000 genes. And so the idea is we want to know what does the inverse correlation matrix look like. This is going to be a 20,000 by 20,000 matrix, where each element corresponds to a pair of genes. And again, our goal, and by the way, just as correlation matrices are symmetric, like if I tell you that the correlation between gene 1 and gene 2 expression is 0.9, then also the correlation between gene 2 and gene 1 expression is 0.9. So too, this is a symmetric matrix. So in this matrix, the inverse correlation between gene 20,000 and gene 2 is 0. And what that tells me is that those two genes are conditionally independent. So we can see that our goal is to estimate sigma inverse. And in particular, I don't really care very much what most of the values of sigma inverse are. I just want to know where the zeros are. Because if I know where the zeros are, then I know where there are edges in my network. So how do we write this statistically? Well, let's let xi be a gene expression vector for the ith sample. So I have tissue samples 1 through 200. I take the ith, and I write its gene expression measurements. So this is a vector of like length 20,000. I have 200 of these, and I'm going to assume that they're independent, by which I mean these should not be like 200 tissue samples taken from the same person. These should be 200 independent tissue samples. And I'm going to make an assumption that they're drawn from a multivariate normal distribution, which is a pretty standard statistical assumption. And in particular, I'm going to assume that the correlation matrix is sigma. And so as we've said, a 0 in sigma inverse indicates a pair of genes that are conditionally independent. So like if I had 3 genes, and here there's an edge between genes 1 and 2 and an edge between genes 1 and 3, but no edge between genes 2 and 3, then that'll correspond to a sigma inverse that has zeros between the 2 and 3 elements. So how are we going to estimate sigma inverse? Well, we have some data. And so the question is, how do we use data to estimate sigma inverse? And so I think it's helpful to think about what we know and what we are looking for. So now I'm going to clarify some of my notation. So sigma is a correlation matrix. But it's not a correlation matrix, actually, that I can just calculate based on the data. It's the population correlation matrix. So it's the correlation matrix for my 20,000 genes if I had access to the entire universe of tissue samples. So it's this unknowable quantity, but we can try to estimate it. So this is like in any statistics class you've taken, this is a parameter that we want to estimate. There's also sigma inverse. This is also unknown. and it's the inverse correlation matrix in the population. And actually, to be totally honest, I don't really care about sigma. I just care about sigma inverse. But we do have some information, and this is what I'm going to call S. And S is the sample correlation matrix. So if you give me an actual data set, like the one that we saw with 200 tissue samples and 20,000 genes, I can calculate S. S is like an estimate. It's like you have 200 people walk into the clinic. You want to estimate the average height in the population. I average those 200 people's heights. That'll be an estimate for the true and unknowable population height. So that's really the analogy here. So S is my sample correlation matrix, and I can just calculate it in Excel or in whatever software I use. What I really want is sigma, or better yet, sigma inverse. So what we want is sigma inverse, and what we have is S. So how can we estimate sigma inverse? Well, so in general, if we have a lot of observations and we don't have very many genes, then actually the sample correlation matrix is a pretty good estimate for the true population correlation matrix sigma. And so if we want to estimate sigma inverse, we can just take the inverse of the sample correlation matrix. So this doesn't sound very hard, except that there's a couple problems. That estimate for sigma inverse is not going to be really satisfactory because none of its elements are going to be exactly equal to 0. So it turns out that this sort of natural approach of just taking the sample correlation matrix and inverting it isn't going to give us an answer that we can use if our goal is to determine where are the edges in the network. And even worse, in the type of data that we see in biology, we unfortunately have many, many more genes than observations. You're never going to have more tissue samples than you have genes if you've done some sort of high throughput analysis. And so we can't even calculate S inverse, because remember I said most matrices have inverses, but not all of them. And unfortunately, S is a matrix that does not have an inverse. So using sort of classical statistical tools, we're not going to be able to estimate sigma inverse. And this is a problem because our data in this context is really high dimensional. We have so many genes relative to the number of observations. And so really basic ideas sort of break down, unfortunately. So in 2007, there was a proposal made called the Graphical Lasso for estimating sigma inverse in order to solve exactly this problem. And I'm just going to briefly describe what the idea here is. So this is an approach for estimating sigma inverse when we have more variables than observations, as we do in the like genomic setting, where we have 20,000 genes and maybe 200 observations. And it basically boils down to, a math problem that probably doesn't look totally obvious, but it involves S, which is, again, the sample correlation matrix. And basically, it's an optimization problem, and when we solve it, we get an estimate for sigma inverse. And in particular, there's this parameter lambda. So lambda here controls the number of elements in our estimate for sigma inverse that are exactly equal to 0. And so it turns out that this is a really flexible method that allows us to use our sort of prior belief of how many edges this network really has. So in particular, if we take a value of lambda that's just equal to 0, then we're just going to get the inverse of the sample correlation matrix, which first of all doesn't have any zeros in it, as we mentioned. And second of all, we can't even calculate it, because as we said, if we have more genes than observations, which we always do in genomics, you literally can't even invert S. So when lambda equals 0, this isn't very useful. But if we choose a positive value of lambda, then we can get an estimate of the inverse covariance matrix, and that estimate is going to contain a lot of elements that are exactly equal to 0. So here's the idea. If we perform this graphical lasso procedure, and we choose this tuning parameter lambda to equal 0, we're getting a very dense network where every single gene is connected to every other gene. But as we increase the tuning parameter, we're going to get a sparser and sparser network with fewer and fewer edges. So like this corresponds to a network in which there's 10 or so edges. As we increase lambda to 10, we now have 6 edges maybe. When we increase lambda to 100, we just have a couple of edges. And when we make lambda really large, we have no edges. So you can interpret this like this. If I tell you, OK, given your data, I'm going to give you the best possible estimate of the relationships among the genes, given that I'm only allowed to assume that there are 1, 2, 3, 4, 5, 6, 7 edges. This is the best way that I can model your data using 7 edges. And if you tell me, hey, Daniella, you can only use 2 edges, then this is going to be the best that I can do. And the idea here is that, as we all know, like in high-dimensional data, it's really easy to overfit our data. So this lambda we can think of as a tuning parameter that tells us how hard we're allowed to fit our data, how hard we can train our model. So basically, the take-home message is that this graphical lasso approach is a really well-motivated statistical technique for which we can actually solve an optimization problem, and on the basis of a given data set, we can estimate the underlying network, and it involves this tuning parameter, which I've been calling lambda, that sort of controls the sparsity of the network. So this idea has been around since 2007, and it's something that has been really well studied statistically, but it hasn't been very much adopted outside of sort of the community of people analyzing genomic data who also have a very strong background in statistics. And instead, what are people doing? Well, people actually just take that sample correlation matrix S and they threshold it. So if you read a lot of papers in which people are talking about understanding like a transcriptional regulatory network, often they're making inference by just taking that sample correlation matrix and they're saying, hey, like gene 3 and gene 9 are really correlated, therefore they must have a direct relationship. And so this is a little bit unfortunate because we understand that that's really not the right thing to do. I mean, it just depends on what you're looking for. If you're just interested in finding genes that are correlated, like sure, look at the correlation. But you should be aware that a high correlation between genes 3 and 9 provides really no evidence for a direct relationship between the two, because as we've seen, it could be an indirect relationship. So instead, people are kind of doing the simpler thing where you just take the correlations, the sample correlation matrix, and you just threshold the elements. So like if a correlation is greater than 0.7, you say that there's a relationship between the corresponding pair of genes. And then people tend to interpret those small, if a correlation is small, they say that the corresponding pair of genes does not have a relationship. So they're kind of interpreting that as conditional independence, even though statistically that's not actually the right thing to do. And again, this is not the right thing to do because that sample correlation matrix is estimating sigma, whereas conditional dependence relates to the inverse of sigma. So why don't more people use the graphical lasso, right? I mean, this has been around since 2007. I'm really partial to it because my advisor was my PhD advisor was one of the authors on the paper. And this is like a really big topic in statistical machine learning. But again, it's not being used that much by practitioners who are non-statisticians. And part of this is due to the fact that this distinction between sigma and sigma inverse is really subtle, right? Like it's really easy to estimate correlations or to compute correlations. You can do that in Excel. You can even do it by hand if you're patient enough. But thinking about sigma inverse is really not very clear. So there's a little bit of subtlety. But there's also another reason, and this is the fault of statisticians, which is that existing algorithms for solving the graphical lasso problem are too slow. So I showed you this optimization problem that corresponds to the graphical lasso. And it's a problem that we can solve. But solving it, unfortunately, is really not feasible. So here's an example. So this is using like the cutting edge software, which everybody uses to solve this problem. So number of nodes, that refers to, let's say, the number of genes in your data set. And then I'm telling you the number of seconds. So the algorithm scales as O of p cubed, where p is the number of nodes or genes. So if I have 500 genes, it takes me 5.5 seconds, no problem. If I have 2,000 genes, it takes me like 3.5 minutes, which is, it's fine. But the problem is that if I have 20,000 genes, it's going to take me 10 cubed as long. And that's really just like too long for most people to wait for their data to get analyzed, right? Especially because there's this tuning parameter lambda that I mentioned, and you might want to try out a lot of different values of lambda to see how the results compare, depending on how many edges are in your network estimate. And this really is not going to work. So this is kind of the situation as of 2011. And so now I'm going to tell you about some of my work in this area. So the first thing that we realized is that it turns out, so remember, the graphical lasso problem kind of has a sort of obscure form. It's this optimization problem. And again, it's really well motivated statistically. But if you kind of just look at it, it's not sort of clear exactly what your sigma inverse estimate is going to look like. But it turns out that there's actually a very simple property of the sigma inverse that you get as you vary that tuning parameter lambda. And the property is that the connected components in the estimate of the network that you get are nested. So what do I mean by this? Well, here's a little example with 7 nodes, and I have 3 values of lambda. And I'm not saying, and of course, as lambda increases, we get fewer and fewer edges in our network, right? So I'm not saying that once an edge is in the network, or once an edge leaves the network, it's always going to be gone. Because we can see some examples where there's an edge that's there when lambda equals 10 and is gone when lambda equals 100 and comes back when lambda equals 1,000. But what I'm telling you is a very surprising result that people in the community didn't realize until recently, which is that the connected components are nested. So like when lambda equals 10, all of the edges are connected to each other through some indirect chain. Sorry, all of the nodes are connected through some chain of edges. So like these 2 nodes are directly connected to each other. And this one and that one are indirectly connected through this one. So if you look at like this indirect chain of edges, every node is connected to every other one over here when lambda equals 10. When lambda equals 100, in contrast, some nodes are just not at all connected to others. So these 3 nodes are totally separate from those 4 nodes. And that's what I'm referring to as 2 separate connected components. So the surprising result is that once you end up with a network estimate with 2 different connected components, then as you increase the value of lambda, these 2 components are going to be disconnected forevermore. There's never going to appear an edge connecting these 2 nodes. Because they've become disconnected, they're going to be disconnected until the end of time. OK, so this is like sort of interesting if you're into this type of thing. So what's realization 2? Realization 2 is that for a given value of lambda, there's an easy way to identify the connected components. So like if you gave me your data set and you said, Daniella, please find out for me, don't actually do the graphical lasso when lambda equals 100, but just check out what are going to be the connected components. So I want to know like which sets of genes are going to be totally disconnected from other sets of genes. It turns out that I could actually calculate that for you really quickly in like 2 seconds on a computer, even for a really large data set, because there's a very simple characterization of the connected components mathematically. And then realization 3 is that we can actually obtain the exact right answer to that original graphical lasso problem if we perform the graphical lasso separately on each connected component. So what does this mean? That means you give me your data set. You tell me that you want the answer when lambda equals 100. So I just need to check out what are the connected components when lambda equals 100. And I can calculate that really quickly. And then solve the graphical lasso problem on each connected component separately. And this, it turns out, can lead to really, really massive speedups in the graphical lasso algorithm. And the reason for these speedups is because, remember, the algorithm scales as O of p cubed, where p is the number of genes. So if I can reduce the number of genes that I need to deal with from like 20,000 to 200, and I just have to do that 100 times, doing something 200 cubed operations 100 times is way better than doing 20,000 cubed operations. So this has really big implications for our ability to analyze genome scale data using this algorithm, because now we can estimate a network in 2 seconds, which achieves our goal of being able to do it while we're on a coffee break. So just to give you an idea, I mean, it used to take 215 seconds to estimate a network with 2,000 nodes. Now it takes less than 2 seconds on this laptop. And it used to literally not be feasible to estimate something with 20,000 genes. And now it takes a few minutes. So like you should get a cup of coffee, but you don't need to take like too long of a break. So it turns out that there's also an interesting fact, which is, I started out this whole talk saying you don't care about the correlation sigma. What you really care about is the inverse correlation matrix sigma inverse. And that's absolutely true. But it turns out that the joke is kind of on all of the statisticians, because there's a really, really surprising thing that we realize, which is that if you actually estimate a network using the graphical lasso, or if you instead just take your correlation matrix that you estimated from your data, just literally correlation the way you saw it in like your AP statistics textbook, and you compare the connected components that you get, those connected components are the same. So the edges aren't the same. As you can see in this example, like within this connected component, there's an edge here. And in contrast over here, there's no edge. So the individual edges aren't the same, but the connected components are the same. So if you want to analyze like a gene expression data set, and you want to infer that there are these sort of separate pathways that are operating independently, and you don't care about which genes are directly related to which genes within a pathway, then you can just go and knock yourself out by thresholding correlations. But if you want to be able to say that like gene A directly relates to gene B, then you should be using the graphical lasso. So I just briefly now want to talk about more recent work which involves joint estimation of multiple networks. And so like the setup that I've been talking about is like you have 200 tissue samples and 20,000 genes and you want to estimate a network. But of course, for a number of reasons, this doesn't correspond to many scientific studies. And one reason is because typically we don't have 200 sort of independent samples from sort of the same distribution. Normally we're doing some kind of comparison, right? So maybe half of our people or half of our tissue samples are tumor samples and half of them are normal tissue. And so then how can we extend this idea of trying to understand networks and pathways to the setting where we have two different conditions that we're comparing? And so for instance, maybe in this, again, in this little example with 7 genes, the normal network looks like that. And then in cancer, things have gone wrong. And we want to be able to determine what edges are the same and what edges differ between these two conditions, because that can really tell us something about what's happening in normal and in diseased tissue. And the idea is that if we can identify these network differences in a rigorous way, then that can reveal fundamental biology. So what can we do in this setting? We have like 100 tissue samples corresponding to condition A, which could be some type of cancer, 100 corresponding to condition B, which could be normal tissue. And we want to make sense of this data. So one thing we can do is we can just estimate each network separately. And we all know how to do that now. We can use the graphical lasso, for instance. But the problem is then the sample size for each network is very small. And we're not really exploiting the information that we have about the data, because we probably think that the two underlying networks are similar to each other, but they have some differences. So if I just estimate network A totally separately from how I estimate network B, it seems like I'm not really taking advantage of what I have. So in contrast, I can just estimate the networks together. I can just throw all of my 200 tissue samples into the software, and I can just get one network estimate. But then I'm really missing the boat, because my whole goal is to really understand how does the tumor network differ from the normal network, and I just can't do that if I just estimate one network for the two together. So the thing that we're going to do is estimate the networks jointly, encouraging most edges to be the same, but allowing for specific edges to differ. And the way we do this is with a formulation called the joint graphical lasso. I know you guys are really interested in the exact form of the math problem, and I don't want to disappoint any of you. So this is what it looks like. And what really, this is not so complicated. It's actually pretty simple. There's some parameter lambda that encourages sparsity in our estimates of sigma 1 of the first inverse covariance matrix and the second inverse correlation matrix. And there's another parameter that encourages similarity between these two inverse correlation estimates. So there's these two tuning parameters, and it's like I can tell my software, I want to estimate two networks that are really, really sparse, and I want to force these networks to be really similar to each other, and then I can see what pops up as different, which edges pop up as being different across the two conditions. Or I can say, I want to estimate these two networks, and I don't really necessarily think that they're that different, because I think that like cancer is really different from normal tissue. But if some of the edges are actually common to the two networks, then I want to allow that to emerge from my analysis. So here's just an example to illustrate the type of thing that you could get. So this is something that a grad student looked at, and this is a data set with 97 tumor and 90 normal lung samples. It's a microarray data set with 18,000 gene probes. And what I'm showing on the left here is the network estimate that you get. So the goal here was to estimate a tumor network and a normal network. So I could have shown you two networks side by side, but then it would be very hard to see the similarities and differences because the pictures would be too small and that type of thing. So instead, I have colored the edges. I apologize if you're red-green colorblind. But the color here of these edges tells you what's happening. So an edge that's in red is an edge that's only present in the tumor tissue. An edge that's in green is an edge that's only present in the normal tissue. And an edge that's in black is an edge that's in common between the normal and the tumor tissue. So what we see is that there are these very tight subnetworks that are totally disconnected from the other genes. And now we know what that's called. That's called a connected component. So there's separate connected components. And we see that some of these little subnetworks tend to be characterized by having predominantly tumor or predominantly normal edges. So for instance here, this is a small subnetwork that's almost all green. And this is a small subnetwork that seems to really only have evidence for it in the normal tissue. And in contrast, there's some other subnetworks that seem to predominantly be present in the tumor tissue. And in fact, there's this huge glob of genes that sort of seem to have tumor-specific relationships, where there might be something going on in the tumor tissue here that's not happening in the normal tissue. So, this, the student showed this to me, and I was confused because there's all of these little lines that might be hard to see from where you're sitting that are just these like short little lines, and I didn't understand what they were. And then he went back and he looked at this, and it turns out, like on a microarray, you can have multiple probes for the same gene. And so then you have to do, well, you can do whatever you want. But one thing that people do is they somehow either choose the probe that looks like it's the best, or they average the probes for the different genes. Or you cannot do any of that, which is what happened here. And so it turns out that these little 2-gene subnetworks, I don't know if you can see these, but for instance, in here there's a little black line, and in here there's a little red line. These are actually connecting 2 probes for the same gene. And so this is maybe not particularly relevant biologically, but it's very reassuring statistically, because what this is telling me is that these 2 genes that are 2 probes for the same gene are conditionally independent of all other genes. So this really highlights the difference between conditional dependence and correlation, because if I believe that these 2 probes are both doing a good job measuring a particular gene, then by definition they're going to be conditionally dependent and conditionally independent on everything else. Whereas if I were thinking about correlation here, I would really have a big glob where everybody basically would be connected to everybody else. So in more recent work, we've been thinking a little bit more. This is with other colleagues from UW, including Sue and Lee in genome sciences, and then a colleague in electrical engineering. And we've been thinking a little bit more about how we can do this joint estimation of multiple networks in a way that's maybe a little bit making more realistic assumptions about the underlying biology. And one thing that we thought about is the fact that if we go back to this picture here, sort of my underlying assumption is that there's a normal network and a tumor network, and they're pretty similar, but some edges might differ. But thinking about it more, that actually might not be the right set of assumptions. So more recent work, we've actually been making a different set of assumptions, where the normal network looks like this, and the tumor network looks like that. And so again, the normal and tumor networks are different, but if you look at this carefully, and it's sort of hard to see in the small example, they're different in a very particular way, where what's really happening here is that there's a gene that was disrupted or perturbed. So if you look at this picture very carefully, and you go edge by edge and see which ones are the same and which are different across the 2 networks, it turns out that all of the edges relating to that particular gene that I'm showing with the blue arrow differ across the 2 networks, and none of the other edges differ. And so this corresponds to the idea that differences between the networks are due to a single gene that is disrupted in cancer. For instance, due to a somatic mutation, maybe. And so the idea is, if you think about why would an edge be different between condition 1 and condition 2, well, it's probably because one of the components of this edge, like one of the genes, somehow got messed up. It's doing the wrong thing. Maybe there's a mutation, or maybe for some other reason, a gene is just in the wrong place, doing the wrong thing, and so all of its relationships with other genes have been totally disrupted. And so we've been working on developing a formulation and also fast software for this formulation so that we can estimate these 2 networks in such a way that the differences between the networks are going to be driven by a particular gene that's been disrupted. And so this is also really more useful scientifically, because at the end of the day, like if you look at your data and you determine that these are the 2 estimated networks, if your software gives you a set of edges that differ across the conditions, it's going to be a little bit hard to understand because, like, what does it really mean that gene 3 and gene 9 are connected in one condition and not connected in another? But if instead what you learn is that gene 3's behavior is disrupted across the conditions, then that's something that you can really follow up on. And you can look in the literature and see if that makes sense and follow up on that gene in a variety of ways. So I'm almost done here. But I want to say that these ideas correspond to, as we've heard, fast software implementations. And right now, the software is implemented in R, which I'm sure a lot of you are familiar with, but R is sort of the language of choice of statisticians, and it's becoming increasingly important. Like, periodically, there will be a New York Times article about how, like, R is the software of the future. So this is great. All statisticians love R. Non-statisticians don't love it as much, but it is really great. And so there's software for the Graphical Lasso and for this joint Graphical Lasso that I've been talking about in R, and you can get the software on this website called CRAN. That's like a repository for R software. And now I'm going to give a shameless plug for a book that I've written with some co-authors that's going to be coming out in May 2013. And so the idea here, so my research area is statistical machine learning, and you've heard about some of it. But the idea is that there's a whole community of people like me who've been, over the past 10 years, developing these really wonderful statistical machine learning techniques, putting out software in R. And unfortunately, it can be really hard to get up to speed on some of this stuff. These are really useful techniques, but it can be hard to get up to speed to them if you're not a statistician, because it sort of requires using this R software that not a lot of people outside of, like, statistics and computer science and engineering are really familiar with. And it also, the way that these papers are published, coming up with these statistical methods, can be pretty opaque if you are not a statistician. And so this is a textbook that presents these cutting-edge statistical machine learning tools for readers who are not statisticians, because really the point of this type of statistical machine learning research is to develop tools to analyze actual real data coming out of various fields. And so this is an attempt to translate some of these tools to non-statisticians. So this is coming out in May, and if you don't like to buy books, the PDF will be available for free online. And that was very generously provided by the publisher, which is Springer. Or I recommend you buy it, though. So I just want to conclude by saying, we all know that network structure plays a really important role in biology. And there's this tendency to use the word network without really thinking very rigorously maybe about what we mean by that. And it is actually really important. This is not sort of like a statistical quibbling point that doesn't matter. This does matter because it affects how we interpret our data and how we understand it. And so network inference is really challenging, and it's an exciting area because it's something that is really useful to biologists, but it's actually also a really active area of research for statisticians, because these huge, massive data sets coming out of biology have led to questions that statisticians had never thought about before. And so there's a lot of really great opportunities for collaboration. And so just in terms of my future work in this area, I'm working on integrating known aspects of networks, because the work that I've been talking about today is sort of assuming that you give me 20,000 genes and I know nothing about them. But of course, and sometimes you might want to take that approach, but sometimes you do know a lot about these genes, and so you'd want to be able to incorporate that known information. And then another question, of course, involves integrating multiple types of data, because I've been talking about genes and where we've measured expression data using like microarrays or sequencing or something. But of course, we have different ways to profile these molecules, and we really need ways to integrate these types of data. So these are all open questions. So that's all that I have, but I'm happy to take questions.
Is there something about the data structure, the dimensionality of the data that defines what the optimal lambda should be?
So that's a really good question. And
Remember to repeat the question.
oh, yes, OK, thank you. So the question was, lambda seems arbitrary. How should I pick lambda? And again, remember, lambda was that tuning parameter in this graphical lasso formulation that controls how many edges I have in my network estimate. So there are a lot of people who think that there should be like an answer to that question, and so there's statisticians who are like proving theorems about what lambda should be for your data. And so the answer I'm going to tell you is my personal opinion, and a lot of people disagree with me. But I think that this is a question for the scientist, because if you think about it, The idea that the true underlying network only has, let's say, like 7 or 700 edges or something, that's just a way that we're simplifying reality so that we can make sense of it, right? This is sort of up to interpretation. So you can think about it like at the 100-mile view, every gene is just operating on its own, and as you zoom in, probably every gene at some point has some interaction with every other gene. So you can think of this tuning parameter as controlling the level at which you're looking at the data, whether you want like the 100-mile view or like the 1 nanometer view. And so there's this trade-off, right? Because if you chose a value of lambda that corresponds to the 1 nanometer view, you'd end up seeing that every gene interacts at least occasionally, at least some tiny, tiny fraction of the time with every other gene. But it wouldn't be very useful for you if I analyze your data and I tell you that every gene interacts with every other gene. And in contrast, the 100-mile view also is not very useful. That would correspond to me telling you none of your genes interact with any other gene. And so it's really something where it's up to you in the level of detail in which you want to see your data. And if it's sort of too complex of a picture, it's not useful. And if it's just a totally vacuous picture where no gene is connected to any other gene, that's also not useful. So I think that there actually is a trade-off that I don't think a statistician can answer. I think that that depends on why you want this estimate of a network. And another thing that I should clarify is that this is like clearly exploratory data analysis. This is just aimed at hypothesis generation and trying to get a picture of what might be going on. And so that ties into the fact that there's no single choice of lambda that would be right for your data. And it's very data specific and application specific.
What happens when data sets are incomplete? I'm thinking more outside of gene expression data, but other kinds of data that maybe you haven't explored yet. How complete do the data sets have to be?
Right. Well, so I would say that in general, for most statistical analyses, if your data is incomplete, you're going to want to do something to sort of impute the missing values. I'm giving you a very vague answer because I don't know exactly why. Oh, I'm sorry. The question was, what do I do if my data is incomplete? And are these methods applicable? So I would say that in general, for most statistical analyses, it really depends on why your data is incomplete. Like if you're, and can you try to impute the values that are missing? And so this sort of, it would really depend on the application. And this doesn't in particular have to do with network estimation, but just for statistical analysis in general. Like if my data is incomplete because like the person who was recording the data, just like every 17th number they were supposed to write down, they just like got distracted and didn't write down that number, then there are things we can do in order to try to impute the missing values. But if the data is incomplete in a really systematic way, like the high values, I know you're not talking about gene expression data, but if you were, if for some reason the high values of gene expression just never got recorded because there's a problem with the instrument for the high values, and so there's a systematic reason that the data is incomplete, then that's going to be a really big problem for any analysis. And you're going to really need to have an understanding of exactly what went wrong in order to try to solve that problem in a really targeted way, statistically. Yeah.
So, thanks, that was really, really nice. It seems like you might be able to test the hypothesis that you can distinguish correlated from truly interacting genes by looking at tissue, or by showing that you never show an interaction among genes that are expressed in completely different cell types. So if one gene is only expressed in neurons and another gene is only expressed in non-neurons, that you would never show an interaction between those.
Right.
Genes. I'm just wondering if you had the opportunity to look at data sets like that where you know something about the distribution of expression of certain genes and to show that something that's biologically implausible doesn't actually show up.
Okay, so the question was, I'm paraphrasing it. I'm supposed to repeat the question, so I'm sorry if I get it a little bit wrong. But the idea is, like, in certain settings, we might really know something about what's happening. Like, there might be a gene that's expressed in neurons, but not expressed in another different type of tissue. And so we could do, like, a proof of concept in order to make sure that we're never inferring conditional dependence between a pair of genes if one of them's expressed in neurons only and the other one's expressed in some other tissue only. And so that's a good point, and we do want proof of concepts. But I think that actually in that case, like, let's say I'm looking at neuron tissue, and like gene A is expressed in neurons, and gene B is just never expressed in neurons. Then gene B is going to have no correlation with any other gene because its expression will just be 0 in neurons.
Well, you take a glob of tissue that had both tissue types in it.
Oh, I see. Oh, that's a really interesting point. I haven't thought about that. So the idea is take a glob of tissue that has neurons and this other cell type in them and see if we can verify the fact that that we don't infer any interactions between the two. Yeah. So I mean, like, again, this is totally like exploratory data analysis. And what we need is ways, this should just be a tool to stimulate the hypotheses that then are drawn from the data in order to stimulate further research. So.
That was my question really is, how have you tested this thus far? Where presumably there are some known biological pathways and others that interactions are known not to take place. And how does this approach behave in those settings?
Yeah, that's a good question. I mean, it's hard to do. It's something that people work on. It's not something that I myself have worked on as much, but some people who are more applications driven and closer to the biology have worked on. And it's very hard. Data is messy, and you're estimating these very high dimensional networks with 20,000 genes on the basis of like 200 samples if you're lucky. And so another problem is that we actually don't really know what the truth is. So like people will often look at the networks that you get and they'll be like, oh look, gene 3 is highly connected to tons of other genes and this is cancer. And I know that in cancer, just doing a PubMed search, like this gene is like highly implicated in all these types of cancer. And so that makes sense. And then of course the question is, if instead of gene 3 it had been gene 39 because I accidentally mislabeled my data, would they still have found evidence, right? It's a little bit hard to quantify the accuracy of the conclusions that we draw, and the problem is because we don't know the ground truth. So I guess my answer, so I should have repeated the question. The question was, how have we verified this? And the answer is, different people have verified it in different ways, but it's hard to get like a really objective assessment of this, and also many other statistical analyses that we do all the time. So the answer to your question also applies to, for instance, clustering, which is used a lot. Like we love to like cluster breast cancer in order to see what the subtypes are, and or conversely, cluster the genes in breast cancer in order to see what are the relevant gene pathways. And there's a similar situation there, which is, unfortunately, we don't know the ground truth, right? There's not an answer written somewhere and we can just check if we got the answer. And so it's very hard. And that's a problem, of course, that we have in a lot of analysis of high throughput biological data.
So if I'm remembering right, and I haven't thought about these sort of things in a long time, all these edges you could get with 20,000 genes would be 20,000 things taken 2 at a time.
20,000 squared divided by 2, a lot. Like basically 20,000 squared, 400 million. So the question, I'm sorry, this wasn't the question yet, but I'm trying to repeat whatever I hear. So if he remembers right, and he does, if there's 20,000 genes, there's a lot of possible edges, and he is correct.
And so as you get these exploding amounts as you go up. Is that still manageable?
So that's a really great question, and I guess I should have started here, right? Like, why do we actually need statistics? Like, why don't we just, like, do something really simple that doesn't require, like, advanced knowledge of statistics? And the problem is because 20,000 squared is too many. If we were just going to go through and look at every single possible edge between a pair of genes, that's too many. It's obviously too many to go through by hand, but it's even just too many for, like, conventional analyses. And what we need to do is make a set of assumptions that under that set of assumptions, we can try to estimate the network. And so here, the assumption that I've made is that the actual network is sparse. The assumption that I've made is that there are not too many edges. So essentially, I'm assuming that though there might be 20,000 squared possible edges, in reality, there's 5,000 or something. You know, the exact number corresponds to that tuning parameter lambda, and that relates to your previous question. But absolutely right. I mean, we're now suddenly in this like high dimensional paradigm where if we wanted to have more observations than possible edges, we would need 400 million people to be enrolled in our study, which is not going to happen. And so we need to really think hard about how we're doing this and make sure we're sort of doing it the right way statistically. And that's 400 million ways we could go wrong in analyzing our data. So, but I mean, I hope you can all sleep at night now.